I just wanted to welcome everybody to the Science Deep Dives today. My name is Lisa Schill, and I'll be your host. First, we have Ethan Perlstein, the founder and CEO of Perlstein Labs, to discuss what is evolutionary pharmacology and how it's spurring research.
This should be on. Does everybody hear me okay? Great. Well, thank you. It's great to be here. This is actually my third Global Genes, so I feel finally a part of this community, and it's not just me anymore. The first time I came here, I had nothing, not even a business plan, just some ideas. Second year, we had just gotten funded, and now here actually with some members of the team. Sangeetha's here, and I think that might be it, but there are a couple other members who are scattered about here. So I want to tell you a little bit about what we're doing at Perlstein Lab. So first of all, the PBC here stands for Public Benefit Corporation. Just want to make that point. Maybe people have heard of this company called Kickstarter that raises money through crowdfunding. They just converted to a Public Benefit Corporation. That's what that stands for. So there's C Corps and there's 501c3s, and then there are these things called B Corps. So B Corps or benefit corporations have to have some kind of a social and environmental mission. So our social mission is the unmet medical need of orphan disease patients, and our environmental mission is to reduce the carbon footprint of drug discovery. So I want to tell you a little bit about what we've been doing the last year and a half since we started. The company began in early 2014. We're based in San Francisco, not too far from UCSF's Mission Bay campus. We're in an incubator, a biotech incubator called QB3 at 953. And I want to tell you about our drug discovery platform for rare genetic diseases. So maybe I can just start with a broad overview of our entire platform approach. So this audience is obviously quite aware of the rare disease space and the challenges of rare diseases, and also the statistics of rare diseases. So the 1 in 12 number everyone has heard, I think, by this point in time, which translates, so that's 1 in 12 people on Earth, so that translates to the United States about 25 to 30 million orphan disease patients. And those patients are scattered across thousands, up to 7,000 different diseases. And the average rare disease has a population size of 6,000 globally. So that's the landscape. And what we want to do is to build an industrializable and scalable drug discovery platform for the rare disease space that can cover those thousands of different diseases. So this is just to remind everybody about that 1 in 12 number. When you actually break it down further and you ask all the patients who have rare disease X, you actually find that if it's a single gene mutation, it's usually never just one mutation. There could be dozens, if not in some cases, hundreds of different mutations that affect that one gene, but it's still a single gene disease. And so this idea is captured here. So if you look, for example, these are single gene diseases that manifest across multiple tissue systems, but a lot of the times it can really be traced down to defects at the cellular level. So in a wild type or normal healthy individual, you would have normal cells. And then if you looked at this particular zoom in of an orphan disease, it's just called orphan disease X, you can see a spectrum of mutations denoted by these different colors. Apologies to the colorblind for the red here. But the point of bringing this up, that there are multiple mutations within this one disease class, is to remind people that this is why we need precision therapy, because there isn't going to be just one mutation, and so there won't be just one deficit or one kind of deficit in the disease. There's probably going to be multiple different shades, and each one requiring potentially its own precision therapy. So for example, in this hypothetical individual, they have a mutation at this particular position in this gene, which is encoding a particular protein. And it results in some kind of cellular stress. So that's kind of denoted here by the cells looking a little bit unhappier than the healthy cells. And this would be another mutation that could be observed in this patient population. And it has maybe slightly more severe effects. And then this patient has a mutation which results in no protein being produced, and so you have cell death. And so you get this kind of continuum. And that's an important theme that keeps appearing across orphan diseases. So our approach is to say, well, let's try to do drug discovery in a way that can be personalized but also scalable. And so we think the way to solve that problem is to use simple animal models for drug discovery. So these simple animal models are yeast, baker's yeast, worms and nematodes, C. elegans, the fruit fly, Drosophila melanogaster, and then the zebrafish. None of these, as you can see, are mammals. So they're considered simple animals, but they're still animals. Even the humble little yeast is a single-celled animal. So we can do drug discovery screens by personalizing these models with particular patient mutations, creating that personal disease that we can then reverse using high throughput screening technology that's been around for quite a while. We would like to eventually engineer the next generation of these technologies, but right now we can use off-the-shelf high throughput screening technologies to run these kinds of drug screens, to look for molecules, small molecules that can reverse the disease. And if we identify these candidates, we can then make sure that they work in patient cells, make sure they work in mice, which are the traditional preclinical models that people use. These animal models are not necessarily the conventional models used by biopharma, even though they are the organisms that drive basic research across the world in academic labs. And ultimately, we want to arrive at precision drugs that are matched for a particular mutation and are most effective for a particular cohort of patients within a broader disease category. So that's the entire platform. And here I'll talk a little bit more about the biology and the rationale for what we call evolutionary pharmacology. So here is a simplified tree of life showing you 5 different animals, including us. So again, reminder, even this simple organism, the budding yeast, which last shared a common ancestor with us 1 billion years ago, even the simple yeast is considered an animal. So if you take a yeast cell and compare it to any one of our cells, essentially, the drawing diagram, the guts of the cell, the various compartments, a lot of the different interactions, a lot of the different pathways, a lot of the metabolism is essentially the same or largely conserved. And that's kind of reflected here in this chart. So if you're looking at a hypothetical sequence that contains a gene that causes a rare disease when mutated, you can see that there might be stretches of that gene that are perfectly intact, that haven't changed at all over a course of 1 billion years of evolution. Of course, there will be other changes that happen along the way, but ultimately, that's what we try to capitalize on and leverage, is this fact that there are conserved, not only conserved genes, but then those genes sit in conserved pathways, which are part of conserved compartments, and so on and so forth. And as a rough sort of rule of thumb, zebrafish would be about 75% at a DNA level. If we compare our 2 genomes, about 75 similar, identical to us. Drosophila would be about 50%. Worms about 40%, and then yeast about a third, maybe 25% of their DNA is identical and conserved to ours. Just as so you keep that frame of reference in mind. And I think the bigger point to emphasize here is that there are actually, of the monogenic disease space, which there are about 3,500 such diseases, a good chunk of those genes are actually ancient genes that have counterparts, maybe not in all 4 out of 4 of these organisms, but at least in 1 out of 4 of those organisms. So when I talk about precision therapy, why do we need precision therapy? Why can't there just be one small molecule drug that would just be the magic bullet for that entire disease? Well, it's because the mutations are not all the same. They don't have the same effect. And so here's an illustration of that. So in the case of an unmutated protein, an unmutated gene, you're able to produce, the cells are able to produce fully folded proteins, which then go on to do their normal function, and the cells are copacetic and everything's fine. This particular mutation causes what's called a misfolded protein. So these are a category of mutations called missense mutations. So they change the spelling of a letter from, say, one amino acid letter to another amino acid letter. And this misfolding issue arises because proteins have to fold up in 3-dimensional space. And if they have mutations that impede that folding process, the protein never gets to its final destination in the cell, and effectively the cells are experiencing a loss or decrease of that function. And then other effects ensue, and in the case of the cellular level, you would get cell stress and an unhealthy cell. Now here's an example of a mutation to the same gene, but has a dramatically different effect because it results in no protein being produced, because it produces, or it is what's called a nonsense mutation, or a mutation that causes the protein to not be fully made. And in that event, that's way worse than not having, maybe this means you have a little residual amount of the protein there, so you can eke by, but in this instance, you have no protein there, and now the cells are dying. So that would be the disease phenotype. Sorry, I keep walking over there even though I have this remote. So here really is how we're able to achieve this industrialization and this scalability across multiple diseases, but also within each disease across many mutations. And it's really enabled by this wonderful technology called Crispercast. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Please don't remember that. I just memorized that 5 minutes ago, but we all refer to it as CRISPR. This is this amazing new technology that was actually discovered in bacteria many years ago. Its true value wasn't appreciated until the last few years. But essentially what this is, this CRISPR-Cas is a combination, it's a large molecular machine. It's a combination of a protein and these nucleic acid sequences that allow one to go into a genome and go to any particular spot they want and produce very focused edits. So genome is a 3 billion, base pair book, let's say, and let's say you wanted to change one letter of one word in one chapter. This CRISPR-Cas is the technology that lets you go in, ignore all those 2,999,000, whatever, and focus in on that one particular base change you want to make. And in theory, although this is still an unoptimized technology, it still has some room for improvement, but in theory, there is no bounds to where you can make changes and what kinds of changes you can make. So in our example, we had a number of mutations here, and so ideally we want to create disease models that are personalized to each of those mutations. So using the CRISPR-Cas system, we can go in and say, okay, for this mutation here, this red allele, we can go in and we can find that there is a corresponding ancient gene in these lower animals. We can then go, using the CRISPR-Cas system, in each one of those organisms, we can go in and direct a specific change and say we want to take this patient mutation and now install it into these particular organisms. And when you do that, you end up recreating the disease. And you have to check that that's the case, and that's what we do in terms of the science that we do. The CRISPR and all that is great, but you have to go ahead, make these models, and then in the laboratory see, do the worms, do the flies, do the fish, do they get sick the way that patients get sick? Not only at the whole animal level, but also, most importantly, at the cellular level, because we think that's ultimately where the disease is arising. So here is the first version of our platform. So there's a lot of room for improvement here, but in spite of being able to take advantage of off-the-shelf equipment, with pretty rapid process time, we're able to go from the chalkboard to actual small molecules that are active in our particular assays. So here in the abstract is how our pipeline works. You have your sick animals. So remember, these are the animals from the previous slide where we've installed this disease-causing mutation, and now these organisms are sick. And so what we want to do is we want to ask, is there a small molecule out there that can reverse that disease state? Again, not only at the cellular level, but also at the whole animal level. And then we can combine, essentially, libraries of these compounds, which we can buy commercially. And we're searching here for new molecular entities. We're not trying to repurpose an old drug for a new use. We're trying to find a needle in a haystack, essentially, a new needle. And so we go through this screening process where we try to identify a particular molecule that can rescue the defects caused by a specific mutation. And so we really can operate this platform with 4 pieces of equipment. We have an instrument that allows us to add the food that these different organisms eat into these what we call these multi-well plates. These are either 96-well plates or 3 to 4-well plates. They're about the footprint of an index card. It's basically like parallelized small petri dishes on these little plastic plates where you can examine many compounds at once and look at many conditions at once. And so we can do these. We can add the food to the plates with this instrument. We can add the compounds from our libraries using this instrument. We can then use this instrument to actually add those animals one at a time, in some cases, to the wells of these different plates. And then we have what we call a microscope in a box. So we can then, at the end of this experiment, visualize in an automated way what was the outcome of this screen. Did we find any molecules that could rescue the disease across these organisms? And in this instance, we would find this red molecule that could then reverse disease, and that's denoted here by the healthy cells. And then stepping back writ large, this is how we imagine the pipeline proceeding. We are a preclinical company, so we need to partner with biopharma, with clinically oriented biopharma companies who could develop our discoveries toward the clinic. But here's how this process would look if you're imagining the kinds of clinical trials our precision drug discovery process enables. So if you took that population of orphan disease patients with that particular disease and you stratified them or grouped them by their different mutations, then you would want to give each of those subgroups a molecule or a drug candidate that is tailored to that particular mutation and that particular set of defects arising from that mutation. And at the end of the day, we can develop these drug candidates into actual approved drugs. And of course, it's not just about the North American or European market. Orphan diseases are a global problem. And so we have that kind of a mindset as well, where our motto is to leave no mutation behind. And so the diseases that we're looking at, the first disease, as I'll tell you in the next slide, which is our kind of lead discovery program, the one that's the proof of concept, is a lysosomal storage disease called NiemannPick. But we are generally interested in what we call these single gene or monogenic diseases, especially the ones that affect children. And so there's a large number of related groups of diseases like this. NiemannPick belongs to the lysosomal or lipid storage disorders. There's about 50 of those. There's also other groups of diseases that affect what we call ancient cell biology. So the congenital disorders of glycosylation. So Matt Wilsey this morning talked about NGLI1 deficiency. That belongs to that category of diseases. The peroxisomal biogenesis disorders like Zellweger syndrome, the mitochondrial diseases, ciliopathies, laminopathies. These are all diseases, if you can kind of catch the theme here, they all involve these ancient compartments of the cell. So mitochondria, remember from your high school textbooks, the powerhouse, the power factory of the cell? Well, there's many diseases that go wrong with the mitochondria, and it turns out mitochondria is a very ancient organelle. Even simple yeast cells have this. And same thing goes for all these other compartments, whether you're talking about peroxisomes, et cetera, et cetera. We think that this model organism approach will be generally useful across the thousands of different monogenic diseases, but we think they'll be particularly useful for these sets of diseases that involve ancient cell biology, because we're using these simple models which may be separated from human beings, hundreds of millions of years of evolution. So we want to maximize the chances of success here. Let's look at these diseases that affect ancient biology, and they also happen to be ones that tend to manifest more early, more severely. So in a lot of ways, following the science here also takes you to the place of most unmet need in some cases. So those 6 slides really capture the process. Now here specifically is where we are with our lead program for NiemannPick. So the way we have set up the company is that we start with a mutation. Well, first of all, we start with a disease and a list of mutations that cause that disease. Then we ask, can we use the worms and the flies and the yeast and the fish to model that disease? And that involves a very simple computational step. In fact, you just go to the internet and you ask, does the yeast or the worm or the fly have a version of that gene? Yes? Great, it's eligible for the next step. Next step would be to use CRISPR, put in a particular patient mutation, and then ask, do the flies and worms and so on, do they get sick the way that the human beings get sick, and especially at the cellular level? If that's a go, then you can progress to the next stage. And so for NiemannPick, we selected that as our proof of concept because for several reasons. So scientifically, primarily, there were already published examples of these worms and flies and so on being useful for this disease. So in 2000, a group published a NiemannPick worm paper showing that you could produce NiemannPick disease in the worm. A couple of years later, someone showed that in the fly. A couple of years later after that, in the zebrafish. But these were all academic researchers, so none of this ended up being translated into any kind of drug discovery modality. So that's where we came in. So for NiemannPick, this is an ancient gene. Again, I said that's kind of one of the criterion we look for this initial set of diseases. So NiemannPick, the NPC1 gene is present in yeast and worms and flies and zebrafish. So it's actually a case of 4 out of 4. Sometimes a gene is not that old. It's only present in the fish, but not in the flies or the worms. So for NiemannPick, we could potentially exploit 4 out of 4 of those models. For the primary drug screen, we ended up using 2 of those models, the worm and the fly. We started out with 50,000 what we call drug-like compounds. These are compounds that have properties that are thought to be quote-unquote drug-like. So if there are any chemists in the audience or anyone has appreciation of chemistry, things like the size of the molecule or its charge properties or other things, people know from looking at the body of approved drugs that approved drugs tend to have certain kinds of properties. So commercial vendors have sell compound libraries that have those kinds of properties, but are otherwise unbiased, right? So because you don't want to go in a screen where all of your needles do the same thing. You would do a screen to find a needle that works for your particular challenge. So for the worm and the fly, we were able to get robust screens to do drug discovery with. And what we saw when we put a mutation into the NPC1 gene of worm and fly is that the worms and the flies had developmental issues. They didn't actually develop to adulthood. And that's kind of writ large what's happening in NiemannPick and a lot of these childhood diseases. We also found at the cellular level that the same kinds of same kinds of liabilities and same kinds of defects were manifesting at the cellular level in these animals as they were in the patients. But more specifically, the way we were actually able to do a screen was to say, well, once we had NiemannPick worms and flies, and we noticed they had developmental challenges, we could then ask, this is now an opportunity to find a small molecule that could suppress or reverse those developmental challenges. In other words, allow these animals to survive to adulthood. So the flies die as larvae, essentially the juvenile phase of the fly. So any animal starts as an embryo, goes through some kind of juvenile phase. If it's not a mammal, they call that juvenile phase the larval phase. Then there is some kind of transition to an adult phase. So the flies never make it to adulthood, so let's ask for small molecules that can allow them to reach adulthood. Worms, they don't die as larvae or juveniles, they just don't reach adulthood as quickly as normal worms. So that screen was designed to ask for small molecules that could allow these mutant worms to reach adulthood at the same time as a normal worm would. Now, from those 2 screens, we were able to sift through those 50,000 compounds and then wind up with about 100 that are really high confidence, that can reverse disease in 1 or more of these 2 simple models, and can also work or have activity in patient cells. So we were able to get from a biobank called Coriell, patient fibroblast lines from different NiemannPick C patients representing different mutational backgrounds. And that's kind of the step in our pathway where we connect evolutionarily from these simple animals to a human disease model. And that's our first indication that we're not just trying to make drugs for flies. We're not just trying to make drugs for worms. As laudable as that might be a goal, we eventually want to make drugs for people. And so the patient cells is that first step where we can start to understand, are we grasping at just random areas of biology, or are we grasping at areas of biology that are really conserved and also have a disease-modifying effect that's conserved? So we spent most of last year, so the company got started, incorporated in February of 2014. The team came together in the spring of 2014. So by the summer of 2014, we had these models either in hand or we're finishing them, and we're making sure that they behaved properly. In other words, making sure that they had these developmental defects, making sure that we could design a screen at the kind of scale to look at 50,000 compounds where we could reproducibly find molecules that reverse that disease state. So the screens actually started in January and took about 6 months to complete 50,000 compounds across 2 different species in parallel. And then over the summer, we started to look at our hits and decide which ones are real hits, because if you get in the drug discovery business, you realize there are things called false positives, things that look like they might be real, but turns out it was not. So you have to go through that process of Q&A. And so we are now at this phase, what we call it in the drug discovery business, hit to lead. So that you start with a drug screen, then you go through a hit validation phase to make sure that the things that came out are actually genuine. And then you go through what they call this hit to lead phase. This is the step in the funnel where you try to identify a molecule that you would one day want to put into the clinic, the so-called clinical candidate. So there's a lot of steps and a lot of hoops you have to jump through to run this gauntlet. And so we began this hit to lead process over the summer, and this begins our transition from the primordial animal models, the ones that we like because of evolutionary pharmacology. We transition from those to the traditional preclinical models, human cells and mouse models or rodent models. So we actually have taken advantage of the fact that not only had people in academia made worms and flies with NiemannPick C, but they also had made mice with NiemannPick C, most importantly. So we're working with a partner that allows us to test our molecules on the NPC mutant mice to see if it can reverse disease there. Now, how does disease manifest in this mice? Similar kind of a theme. Those mice die prematurely. They do make it to adulthood, but they don't live a normal lifespan. And so the test for us is to ask, can any of the molecules that we found that can reverse developmental disease in the lower animals, that has effects in the patient cells, do they actually also extend life and reverse disease in the mouse model? And if we can find that, that can then allow us to identify what we call the lead candidate. This will be the molecule that goes through the next hoop of the drug discovery marathon, which is the lead optimization phase. This is where you do a bunch of chemistry to take the molecule and start to change it and make it more potent, make it more stable, make it more selective, make it have properties that are more quote-unquote drug-like. And what we hope to do over the course of 2016 is to complete these mouse validation studies so we can essentially show that our molecules have climbed the tree of life from the roots up to the mice, and which to us gives confidence that we're dealing with potentially a mechanism of action that hopefully is new. And in fact, we have found a new mechanism of action for NiemannPick C. But most importantly, we want to find a mechanism that works in the animal model, the traditional preclinical animal model. That's what's going to get buy-in from our clinical partners. That's what's going to get buy-in from our investors. And I think that's also just going to get buy-in for ourselves, because again, we don't want to make drugs for lower animals. We want to make drugs for people. And so hopefully we can, through the validation in these mouse models, we can identify that lead candidate, do the improvements chemically to find a version of that, a derivative of that parent molecule that's better, stronger, and so on, and then file for or apply for what's called orphan drug designation. And that to us is sort of going to be the key milestone in this particular project. And we don't aspire to be a fully integrated biotech company. What that means is we don't want to go from concept to approved drug and post-marketing, sales, approval, surveillance. We don't want to do everything because we don't think that we can do everything as well as, for example, on the clinical side, what Big Pharma has spent decades getting good at. What we really want to do is get really good and nimble at the discovery and preclinical side of things. And then we get up to that preclinical, clinical threshold, and that would trigger a partnering event. And then we let our partners focus on that one disease program and take that all the way through. And then we go back and iterate on the next disease. And as I said, there's 3,500 of these rare diseases. Not every single one of them is going to be able to be modeled in our system, but we're not looking for 100% efficiency. Even if we're only able to have an impact 10% of the time, if we take 3,500 attempts, that's still 350 diseases where we might have established drug programs. And I just want to sort of, before going to Q&A, I just wanted to point out this is obviously not me. This work I've shown is a product of an amazing team that has come together in the company. Sangeetha's in the audience here. She's, among other things, is our sort of worm expert. So if you had any questions about nematodes, ask her. Tom, among other things, is our chief fly scientist. And Nina, among other things, is our chief patient cell scientist. We have a group of advisors that's helping us in this journey since none of us have ever made a drug before. We may not have the profile and the demographics of a typical biotech company. This is my first company. I didn't imagine ever being a CEO of a drug discovery company, let alone an orphan drug discovery company years ago. I was sort of on the academic track and left that world to pursue this. All of us were former postdocs, so we've had not only a PhD training, but then essentially a second PhD training in another subject. So we definitely have the scientific bona fides. We haven't made a drug before, but that's why we rest on some of our advisors who have been in the business. So we have a chemist who's worked on what they call medicinal chemistry. That's the improvement of molecules from their screening hit to the thing that you want to put into a person. He's got over 20 years of experience, so he's helped us in this process. We have a business development CEO guru who I have a call with every week. She has her own biotech company that's focused actually on cancer drug discovery, and so I can get the wisdom of someone who's actually been a CEO before, which has been great. And we also actually have a virtual lab manager / CFO who takes care of all of the ordering and all that stuff that allows us to just focus on our science. Matt Wilsey is an investor and a board member, and Linda Avey is another board member as well. So that's our team. Happy to field any questions. Please, thank you for giving me the opportunity to speak.
Tracy Van Houten from the Bat Disease Group. Ethan, first question I have is how do patient groups and patients fit into the equation? Are we a funding partner? Do we have an interest? Is it just naming the mutation you go after? How do we fit?
Yeah, great question. So right now we're focused on our lead program because that's what's going to establish our business and hopefully provide us the credibility to then raise more capital, but also the ability to take this disease program further. But going forward, I think the one role that we kind of are imagining for patient advocacy groups is sort of to enter at the kind of ground level right after the diagnosis. In fact, I was just talking to a mother at the lunch who had just completed the diagnostic odyssey, now has a gene, but now is like, what do I do? And so we want to shorten that distance between diagnosis and discovery. And so one way we can imagine working with patient advocacy groups, both collaboratively, but also with an intent to raise funds to start kind of a pilot project to begin to make that transition from diagnosis to discovery. So for example, can imagine this part of the process where we can say, okay, we know that this is the disease that we're interested in creating models for. We know what mutations we want to model. So maybe one way to go forward is to raise some funds, maybe through crowdfunding, maybe somewhere on the order of, say, $25,000, $50,000, and say, we want to now create a worm, fly, yeast, fish model of your disease with your mutation of interest. And if it's several mutations that are of interest, we could figure out ways to either find some kind of consensus mutation or at least some initial representative mutation that would make everyone happy to say, we've got a starting model. So that's one way we imagine getting the process started, is to say, can we work together to raise funds so that we can build these models? We would want to use the models to do drug discovery. And initially what we could say is, on that kind of a budget, we could say we could do an initial repurposing kind of screen. We could take the collection of FDA-approved drugs and do a quick screen and say, not only to kind of calibrate our model, but just to ask, can we serendipitously find anything that might be useful either as a tool compound or maybe even as something we want to repurpose for real. But that could be a kind of initial starting way to collaborate with patient groups where we don't have to depend on a biopharma partner to start a drug discovery program, where we would want to work with the patient groups because they would know the problem problem best, but in return, we would want to then say, you can take these models and now distribute it to your group of academic researchers who are doing basic research. We would just ask that you don't give it to someone who's going to do the same thing we're going to do, which is to do drug discovery. We just want a little bit of a head start. But ultimately, if you pay for the model creations, they're your models. And so that's one way we're imagining being able to work with patient groups from the outset. Does that answer the...
Yeah.
Cool.
Hi.
Hi.
Cindy Luxhoy-Han. I'm with the Alagille Syndrome Alliance. And we know that our disease is on the JAGAD1 gene. It's in the Notch signaling pathway. We know that there's a huge range of mutations, deletions, translocations, all of which are causing this disease. I guess my question is to be able to look at those specific different mutations that are occurring. Where do you get that basic information from? For instance, we have, there's a hospital that has, or a Children's Hospital of Philadelphia, they've been collecting blood samples. They have a huge database, however, that's proprietary. And people have been consented just for their studies, not for other studies. So how do you get to that information?
Yeah, it's a great question. So, I mean, it's another reason actually why we started with something like NPC, NiemannPick, because you could look at some of the public databases like ClinVar and others, or even just looking at the published literature, and you could very quickly start to gather a list of mutations. And not just a list, but also some frequencies, right? How common is this one relative to that one? In cases where the database is proprietary, or maybe this is, this was a case of, undiagnosed group and now falling into a diagnosed category, I think that presents a bit more of a challenge. I mean, for us, right now, we're obviously just limited to the publicly available databases for mutations, but we can imagine if it were, especially if we were entertaining a possibility of working with a patient group to start building their models from the beginning, then presumably they would tell us what the mutations were. But I think that is a challenge if the database is proprietary. And it's not something we've thought about too much, unfortunately, at this time, just because we've been able to rely on diseases where you can access that information. But I appreciate you pointing out that challenge, and we need to figure out a way to how do you access that information if it's not publicly available?
Do you ever use the individual patient organization's registry as a source of information?
Not yet. So again, we've focused really most of our efforts on NiemannPick, where again, the patient advocacy group infrastructure has sort of already been well established in the sense that there were not only multiple groups, but there were sort of legacy older groups, younger groups. So we felt like we understood that landscape a little bit better, but going forward, especially thinking about onboarding new diseases, I think we would obviously start with the patient groups and kind of go from there. But with NPC, it was just this case of so much was already known, so much had already been de-risked, that was just the logical place to start.
Hi, so I'm PJ Brooks from the Office of Rare Diseases Research at NCATS, and scientifically I understand all of what you're saying, but the part I don't quite get, and maybe I just missed it, is what does working with the model organisms give you as opposed to doing the drug screening on the patient cells?
Right. So great question. So we think there are multiple reasons why doing a drug screen with a whole animal first versus a cell line is better. So for one thing, an animal is an animal. So you have physiology, you have tissues, you have the communication between tissues, you have all that complexity, which you can never get in a cell. You also have the fact that in model organisms, you have these amazing genetic toolkits that have been built by academic researchers to interrogate pathways, to do target ID. So even though these are what they call phenotypic screen, and that's a term of art for people who are not familiar, this is a way of doing drug screens where instead of trying to take a protein that you've isolated physically and put in a test tube and try to find a drug to inhibit it, phenotypic screens are cases where you have a cell or an animal that has some phenotype, like it's sick or dying, and you want to try to reverse that. Now, initially, you don't know how did you reverse it, because it could be acting in any pathway or any target to do that. So the fact that we're using model organisms that have all of these genetic toolkits with them means that we can now approach the target and pathway ID question, I think, much more tractably. The other advantage, we believe, of whole organisms, simple but whole animals versus cells, is that we believe that the whole animals provide a faster path from diagnosis to discovery. So to build a screen for a cell line, you're probably gonna have to characterize that basic biology, understand what's going wrong, what do I have to fix? Maybe you have to do some genetic engineering, build some reporters, build some of this. With our system, all you need to do is put the mutation there, and if the animals die or get sick, that's it. You can treat it as a black box because you can worry about the target ID after the fact. And we also think that doing whole animals first instead of cells first. Remember, we use cells, we just use cells second. So we think that the real reason why we think we're potentially really excited about the whole animals is that we think the whole animals might actually result in higher quality leads going into the further drug development processes. In the sense that even though you're talking about animals that don't have livers, where most of the drug metabolism happens in a human being, worms and flies, they still have to process those molecules. They don't have livers, but they face the challenges of dealing with foreign molecules coming into them. So they can break down drugs the way we can. They don't have the liver, but they have those enzymes that do that. So we think that when you do a whole animal screen first, you weed out a lot of the crap compounds that come from cell-based screens. Because a cell-based screen, again, you're talking about a cell, which is just one cell. You've got many, many cell types. The disease is many, many different features, and you're just looking potentially at one aspect of that disease biochemically that you want to reverse. So we just think there's more opportunities for you to miss stuff, and also more opportunities if you're dealing with a cell line, more opportunities to kind of fall in love with a particular model of the disease or a particular hypothesis about what's going on, because it's a case of that street light. You see what's under the street light. So if your cell-based assay is about looking at the activity of protein X, you're missing everything else. Whereas in our case, we happily treat the animals as a black box because all we know is we have, are they dead? Are they alive? And then we worry about all that rest later.
Can I ask one other?
Yeah.
Can you explain what a public benefit company is as opposed to other kind of companies?
Yeah. So if you think about the spectrum of incorporation, you've got C Corps here, for-profits obviously, 501c3 nonprofit over here. B Corp would be just right here. So the majority of states, actually I think the 3/4 of states, have passed some version of a benefit corporation law. And all that law really says is that you have to add some extra language to your articles of incorporation that say that the company will balance maximizing shareholder value with a social and environmental mission. It's just a couple extra sentences in the articles of incorporation. It doesn't take much to be a benefit corp. Now, you have to follow through, obviously. And because it's a new concept in the corporate sense, not many companies, I don't think any companies in biotech are public benefit corporation. Most of the public benefit corporations that are B Corps that you see, Kickstarter, Etsy, these are all consumer-facing marketplaces. Bakeries, breweries in some towns it's a favorite thing to do. It's non-biotech, so it's a new thing that we're doing. And again, it doesn't really differentiate you other than that couple extra sentences in your articles. Like there's no tax advantages, there's no investor advantage, none of that there is yet. But we think that's going to happen because the broader movement for people wanting to see either a more humane capitalism or whatever, a different form of capitalism that, especially in this area where people talk a lot about patient interests and so on, but then just this week in the news we heard a story about a drug company raising prices. And so the public benefit corp is this idea where hopefully you can do a socially responsible business and have it be as actually part of the company DNA, not just something you kind of say you're going to do. So it has to be something we grow into, but it is the framework we built for the company.
Ethan, I'm curious about the size of the compound library. And
Yeah.
Is that driven by science, by economics? And would you expect a vastly different number of hits if you used 1 million or 2 million compound library? And once you go from lead to hit to lead, do you rescreen with more flavors of gray than?
Yeah, so the 50,000 was chosen strategically to try to satisfy a lot of different constraints, economic, scientific, and so on. So scientific, we knew that we needed to have at least a certain number, because if we had too few, we were worried that we just would miss something, especially because we thought that we would be getting hit rates that would be well less than 1%. That's another big difference between whole animal screens and either cell-based screens or in vitro protein screens. The hit rate is much, much lower. It's much more stringent to get a hit in these. So we see hit rates that are closer to almost like 0.2, somewhere between 0.1 and 0.5%. So 50,000 was, with that number in mind, 50,000 was there to say, let's at least hopefully find something. Anything more than that, given how much we raise and our constraints, screening anything more would have just been logistically very difficult. Now, the plan is to scale up the size of the library. Probably not to 1 million, probably more like 150,000, because we anticipate the hit rate to essentially be constant. I mean, the hit rate is somewhat a function of what kind of molecules you're screening, but if you really are screening unbiased compounds, then Yeah, so that's kind of the rationale but we always are looking for novel molecular entities. That is the key. So these libraries that we're looking at, the starting material, the parent compounds are all open and non-patentable, right? So it's about finding the hits and then doing some medicinal chemistry. So, and you're finding the shades of gray. So the analogy would be, first you do the primary drug screen is a needle and a haystack. And then you throw that needle into another haystack of needles. And then you try to find a more shiny one. And that's the thing you put into a person. So that whole process of making new analogs, that will certainly happen as we're doing now, but that's not going to be more than, say, a couple hundred to maybe 1,000 or 2 analogs of that exciting compound.
Hi, I'm Cindy. I'm with the National Gaucher Foundation, and I also have type 1 Gaucher disease. I'm wondering, I came in late, so excuse my ignorance, and my question might be very off base, but with what you're doing with the whole animal, we have 5 treatments for type 1 Gaucher disease. We have 0 for type 2 and 3 because of the neurological aspects of the disease. Could something like this help in finding a treatment that crosses the blood-brain barrier?
So maybe. I mean, in theory, yes. So if you do any kind of small molecule drug screen, if you're dealing with a neurological disease, you obviously want blood-brain barrier penetrability. And blood-brain penetrability is a property of many small molecules. It's not something that you can just engineer from first principles. You have to filter for them. But the way we set up our screens, we absolutely think we could capture small molecules that are blood-brain penetrable. I mean, just again, think about it from the perspective of just the fact that you're dealing with an animal. Even a fly has multiple tissues. Obviously, it has a brain, it has a head. So in the case of our screen, we're dealing with the juvenile forms, which are basically maggots in the colloquial speak. We call them larvae because it sounds more clinical, but it would be a maggot. So they burrow around in the food. And the way we've done our screens is that we put those 50,000 compounds, we dissolve each of those 50,000 into one well of a 96-well plate full of this molasses-based food that these larvae eat. So those larvae have to eat that food, it gets into their gut, and then it goes from their gut to other tissues. Now, the larvae don't have a brain or anything, but the fact that even these simple animals have almost a digestive system, they have to get molecules going from tissue A to tissue B, there are questions of oral availability, there are questions of excretion, all these other things, even though it's not quite like our situation, it's this primordial form of it. So we absolutely think this platform is compatible with finding small molecules that are blood-brain penetrable. That's a mouthful. Yeah, sorry.
No, I missed somebody who had their hand up earlier. What?
I had a question, but he answered it.
Oh, okay. I actually have a question for you.
Cool.
Do you work with any gain-of-function mutations?
Great question. So yeah, so what we're looking at in all these diseases that we were talking about, and of course like this chart here is really emphasizing the fact that these are all various forms of loss of function, so recessive diseases, but there are these dominant or gain-of-function diseases. We haven't looked at those yet, just because these are a lot easier to kind of understand the biology. You're missing this gene, you're missing this function, so either if there's any residual function, you can either potentiate it, or if that function's all gone, you gotta find a bypass around it. Whereas with a dominant, it's sometimes harder to just dissect the basic biology to understand what is the gain of function, what is this new function. You have to characterize that. If it's a loss, it's a loss, and it's just a matter of degree. So we definitely have a dominant on the radar, but it's a little bit more challenging biologically, so we're starting with the recessives.
So, it's endless, right? The realm of the possibility.
Yeah.
But I'm sitting back here thinking, okay, great, and there's X number of candidate rare disorders. So my question is, what's your protocol to establish criteria, assess? I'm assuming that you're trying to profile a higher probability for success for the outcome. Mm-hmm. And then ultimately, I'm assuming also that you have a model that allows you to go through the process where you say, it just doesn't make any sense to go forward, cost-wise.
Right.
And so cost, if one of the patient advocacy groups would be interested in trying to raise the funds to at least to go through the initial stages, what does that look like?
So I think the cost structure is something as I was saying before. I mean, you can imagine the progression going as follows. Say, raising between 25 and 50K for a 3 to 6 month project that would involve taking a mutation, or set of mutations, and using CRISPR to install those mutations into those models, and then the end product of that are going to be models. These organisms that have, you know, whatever diseases, and they're going to have different flavors of that depending on the mutations you put in. So so for 25 to 50K in 3 to 6 months, you would end up there. And these would be for multiple models. The yeast are the cheapest to make. You know, worms and flies are a little bit more expensive and a little bit longer to make. Zebrafish is actually still a bit challenging just because it takes a long time. May take you half a year to more to get that. But for yeast and worm and fly, 3 to 6 months is a totally reasonable time frame, not only to generate these models, but to validate them, and then also to potentially see, do they have disease phenotypes that you could then screen and try to reverse. So that first phase would be 25 to 50, 3 to 6 months, and then you would follow that with an equal, you know, another 25 to 50K and another 3 to 6 months to actually do a drug screen. Now, we did a 50,000 compound screen, which is, you know, pretty arduous, but we probably, in this instance, we would say, let's just start with an FDA-approved library, several thousand compounds, and let's just see, not only to calibrate the models in some way, just to see that they behave appropriately in the screening context, but do we get lucky and find something? And if we did, then we would send it to our friends, you know, over Recursion, and maybe they can characterize it. But I think the idea is that we would have this kind of pilot program where we create the models and then do an initial kind of pilot screen. And then the question would be, what do you want to do next? Because for our purposes, you know, as I said, you can treat the animals as a black box. You give them the mutation, do you get the disease? You don't have to worry about the details until a little bit later. But, you know, there's a lot of basic biology that needs to happen that we can't do. So if those models were created, those with crowd funds, then those will go back to the patient group and say, here, you're building a just like Matt Woolsey was talking about, go out there and build a consortium of basic researchers, build your brain trust. Well, now you've got some models that they can go work with right out of the gate. And so that's kind of how we're thinking. And in terms of the whole landscape and how we would progress, we have personally identified about 250 diseases, 250 childhood monogenic diseases, of which lysosomal storage diseases are about 50. And that's a set that we'd like to work with because we think that there's a higher probability that that disease biology is going to be conserved. But of course, it's a statistical process. So some amount of the time, it's just these models are not going to pan out because the biology is complex. But because we use multiple models, that is a bit of a hedge. So if one of the models turns out to be, you know, give you nothing or something confusing, then maybe you look for consensus among the remaining models. But if you only had one model and it was one model or bust, that's where I think you get this fragility and this, you know, the lack of robustness in the process. Because then if that model or theory is wrong, then your program's bust. But if you have multiple ones, you could still survive that. So that's how we're thinking about the progressing forward.
Ethan, if our different diseases have some of these models already created, which we do in bat disease, but I'm not exactly sure of the specific mutations,
right.
Are you willing to collaborate with those academic researchers that have built those, or do you want to build everything yourself from scratch?
So,
and I totally get wanting to do it from scratch.
Well, we would want to do it from scratch if the mutations we were interested in modeling weren't available by the existing model. We'd be perfectly amenable to collaborating with existing models. The only wrinkle there is it turns out that that's maybe more complicated in practice than in theory because of material transfers agreement, depending on who made that and who funded it. Getting it out of a nonprofit into a commercial entity, all of those things take time and often, you know, sometimes it proves to just drag out forever. And sometimes also, you know, it comes with, you know, conditions. And so sometimes it's just simpler and more straightforward for us to just recreate the model ourselves. Besides, we would be using CRISPR to make it. Chances are these other models may not have been may have been using other sort of previous genetic engineering technology. So for a lot of reasons, it may just be simpler and cleaner for us to make it from scratch. But if you've got something already that we can just get our hands on and say, can we just see if this produces, you know, disease phenotypes that we can screen for? That's something I think, you know, because that would just be sort of for internal R&D, which is these MTAs are usually really easy about. But if it's like, well, what happens if these materials are useful for something you're going to commercialize? That's where it can get kind of sticky. And depending on the organization, so like Stanford is an institution that has a very kind of uniform and consistent policy that is very pro getting stuff out there. But I've dealt with some institutions where it's a nightmare. And, you know, like 6 months go by and you haven't even gotten your preliminary, you know, documents signed. And it's just, why would we can't waste that kind of time.
Any more questions? Well, thank you so much, Ethan.
Thank you, everybody.
Really appreciate it.